And also, if you mark the GC content, it's also showing that this pattern, this LGT or HGT, happens much more within the similar GC content, but not for the higher differences on GC content, which is also reasonable. If the two genome has a very different GC content, and you transfer a piece of DNA from here to another places, if the GC content is quite different, then you will be less likely to retain this new piece of DNA. So it's much easier for the HGT happen to have the similar GC content. Of course, there are some other cases, and they also actually show some of the very nice example to explain why this happened. And also, here is another thing showing the different habitat of those bacteria, showing if they are from the plant, is a plant pathogen, or is a marine bacteria, or from human, or is fresh water. And basically, it tells you some kind of pattern here is that the one with similar pattern or similar habitat will have a higher frequency of horizontal gene transfer, which is reasonable. And why are they doing this kind of study? And they want to see if there is any barrier to constrain the horizontal gene transfer, to prevent the horizontal gene transfer. And we can see from the slide here and the previous slide, the thing with these similar genomes will have less probability for the horizontal gene transfer. That could be according to the codon usage adaptation. If it's a distantly related genome, it will have a quite different codon usage pattern for the one here, from here to here. So it's unlikely they could have easy horizontal gene transfer, which is reasonable. And we already mentioned the GC content problem, which is reasonable. So this means that the genome property will prevent some of the horizontal gene transfer to happen, even though in bacteria it's very easy to have a horizontal gene transfer. There is still some barrier and some constraints for it's not every bacteria can have horizontal transfer. There is still some pattern here. So this is some of the new tool they divided. So okay, well, I already mentioned some of those. And basically, the horizontal transfer will only happen in a certain criteria, in a very similar codon usage or similar genomic group. And also, they also found out that the lateral gene transfer is much more frequently for those metabolic genes other than those they call the central reaction genes, which means the replication, translation, transcription, those kind of genes are very less likely to have horizontal gene transfer. But those genes had metabolic functions. They call it's the surface reaction, and will have much higher frequency for the horizontal gene transfer, which is also reasonable. But their data clearly show that they support this kind of data.